Okay, so in this video we're going to talk about matter in different types of states. So earlier we talked about atoms, and atoms were the fundamental building blocks of our world. And when atoms are chemically bound together, they can form either molecules or compounds. So a molecule technically is two or more atoms that are bonded together, and it's more of a general term. But a compound is a specific molecule that has two or more different types of atoms. So an example of a compound would be something like glucose, which is 6 carbons and 12 hydrogens and 6 oxygens. And this we would call a compound. This differs than just generally molecules, which could also refer to molecules that are just similar atoms, like H2 or O2. So you have two oxygens that are bound together or two hydrogens bound together. We wouldn't call these compounds, but we could call them molecules. Now, mixtures are just basically where you have different types of matter that are all mixed together, and they're physically intertwined or mixed. There's 3 different types of mixtures that we'll encounter in biology. That includes solutions, colloids, and suspensions. So a solution is when solute particles are very tiny. They don't settle out or scatter light. So because these are very tiny solutes dissolved within this particular solvent, they don't settle, so you don't see them kind of building up on the bottom of this bottle. And nor do they scatter light, so it's just going to look like clear water, even though there are solutes dissolved within this water. Now, a colloid is different here because the solute particles are larger than they are in a solution, and they do end up scattering light. So that's what we see here with the example of Jello, how light is scattered, which makes it look different than just pure water. But a colloid has solutes that don't settle. So what you find then is that the solutes that you use to make Jello, which is gelatin, they don't just kind of dissolve, they don't just settle at the bottom of your Jello tray. Fortunately, it all stays mixed, and you get this particular example of a colloid, which is this type of mixture. Now, a suspension is where solute particles are very large. Because they have so much mass, they are able to settle out, and they may also scatter light. An example of this would be some of the red blood cells that are dissolved within your blood. And this is an example of a suspension. So that as long as your blood's moving and constantly being kind of turned around and mixed, red blood cells stay mixed within blood itself. But because these red blood cells are so large, they have a tendency to settle at the bottom of a test tube if you let that blood sit there for a while. This is an example of a suspension. So there's lots of solutions that can, there's lots of mixtures that can be a suspension, and blood is one of those. Now, this typically wouldn't happen in your body, but this can happen in a laboratory setting if blood sits out too long. The cells can separate on their own just due to mass. Now, solutions are just a homogeneous mixture where you have an equal and evenly distributed mixture of solvent and solutes. The solvent is the substance that's present in greatest amount, usually a liquid like water, and a solute is a substance that's dissolved within that solvent. So typically present in smaller amounts. An example of this is blood sugar, where glucose would be the solute. But blood plasma, which is mostly water, would be the solvent that dissolves that blood glucose. Or if you think about like Coca-Cola, like the water in that Coca-Cola would be the solvent, and the sugars and the other types of flavoring agents and maybe some ions in there would be the solutes that are dissolved in that solution. Now, true solutions are usually transparent. So we say that air, which is a gas solution, is transparent. Salt solutions that are dissolved in water should also be transparent. Same with sugar solutions. Now, most of these solutions in the body are true solutions of gases, liquids, or solids that are dissolved within water. An example of this, you might find some gases within your airways of your lungs, or your body liquids are often solutions, or sometimes there could be some solids that are dissolved within the watery environment of your body. Now, in terms of concentration of true solutions, there's 3 common ways to express concentration. We can talk about concentration as the percent of a solute in total solution, and this is where we talk about like the parts of a solute are in 100 total parts. So when you start talking about like parts per million or parts per billion, they're just talking about the percent of that solute in the total solution. So the solvent is usually water, and so you might say that there's 10 parts of salt and 90 parts of water make a 10% salt solution, because 10 out of 100 parts is 10%. Now, you can also express concentration in terms of milligrams per deciliter. So mgs per deciliter is often a common form of notation to describe concentration, especially in healthcare, where 1 deciliter is 1/100th of a liter, because deci means 100. An example of this is where you can measure glucose. It's measured in the order of milligrams per deciliter. So you're looking at the number of milligrams of glucose. So 1 milligram is 1/1000th of a gram, and a deciliter is 1/100th of a liter. So this is just an example of a particular concentration of sugar within your blood. Now, molarity is the third notation of concentration, and it's denoted as a big M here. This is the total number of moles of a solute per liter of solvent. So 1 mole of a compound is equal to its molecular weight. And so a mole here is determined by what we call Avogadro's number, which is 6.02 times 10 to the 23rd molecules of that substance. Now, this is just a standard number we use to determine molarity. So if you know the molecular weight of a substance, and you know that 6.02 times 10 to the 23rd molecules or atoms of that substance per mole, then it's pretty easy to calculate molarity here. So what we find then is that glucose, C6H12O6, has a molecular weight of about 180 atomic mass units. So 180.12 grams of glucose added to enough water to make a liter is 1 molar of glucose. And you might find that certain substances are expressed in units of molarity. But in the body, a lot of molarities are very small. Because you're not going to dissolve 180 grams of glucose within 1 liter of water. I mean, that's a lot of glucose. So in reality, in our bodies, molarities are more denoted in millimoles, which is on the order of 1/1000th of a mole. So 100 millimoles, 1,000 millimoles is 1 molar. So you might see that there's a lot of like M, little m, big M notations for molarity when we talk about concentrations in the human body. Now, colloids, also known as emulsions, are a heterogeneous mixture, which means that there's particles that are not evenly distributed throughout that mixture. So this is where you can see large solute particles in solution, but these don't settle out. And this can give a solution a more cloudy or milky sort of appearance. So some undergo sol-gel, solution to gel transformations. An example of this would be like where Jello goes from liquid to gel, or when the cytosol of a cell is also a sol-gel type of solution, where it's typically gelatinous, but it can liquefy, and vice versa. Now, a suspension is also a heterogeneous mixture that contains large visible solutes that do settle out. So an example of this would be where you have water and sand, where you might have that sand mixed within water as long as it's turbulent, but as soon as that water and sand settle, the sand eventually will fall to the bottom of that vessel and then form a suspension. Now, blood is considered suspension because if it's left in a tube, those blood cells will settle out over time. Now, the three main differences between mixtures and compounds is that unlike compounds, mixtures don't involve chemical bonding. But mixtures can be separated by physical means. So like straining or filtering can help to separate compounds. Mixture. Otherwise, compounds can only be separated by breaking their chemical bonds. And mixtures can be heterogeneous or homogeneous, where compounds are always homogeneous because you're talking about that's a particular type of molecule made of lots of different atoms.